It's my pleasure to introduce Andrew Yang, Dr. Andrew Yang. Andrew is an incoming Center Faculty Fellow at UCSF, developing molecular tools to study brain vascular and immune contributions to brain aging. He got his PhD from Stanford in Tony West's lab and grew up in LA. Now, Andrew, feel free to take over and start your presentation.
Hey, thanks for having me. I'm excited to be here, and we can make this conversational. I'm excited to share our recent work. It's going to come out in about a week or two in Nature, but it's really just providing a first single cell atlas for the human blood-brain barrier, and in the context of healthy, but also in Alzheimer's disease patients. And the motivation, as it is for many, is the tragic toll that is brain aging and neurodegeneration. I'm personally fascinated by how brain health and homeostasis that's maintained so efficiently throughout most of our lives can just so dramatically fail, right, once you get to the end stage. And it's really hard to capture what that means in words, but there's this beautiful series of self-portraits from William Utermel in Philadelphia as he went through Alzheimer's disease. And you can kind of see that it's a kind of slow death of the self where you slowly unravel and lose your sense of meaning and memories and even identity. So how do you even begin addressing something so complicated, right? And for me, at least, my background was in engineering, and you're always taught to look at the boundary conditions or the edge effects where a lot of the interesting biology or action happens. And so if you look at the brain, it's encased by a very unique structure, the blood-brain barrier, that's critical to establish, but then maintain that brain homeostasis. And one analogy would be to think of this blood-brain barrier, or BBB, like a home, right? You have the residents of the home, and it's important to have these physical walls, no doubt. But to keep the folks in the home happy, you also probably need to have doors and windows to let the groceries in, take the garbage out. Nowadays, you have thermostats to control temperature and heating and sensors. And you also have communication, right? You have Wi-Fi signals and cellular. And I personally believe, and this is what our new lab hopes to decode, we think that's going to play a very important role in shaping brain health, right? And historically, the BBB has been defined by that namesake barrier property, right? You inject the dye like here, and you can see the dye extravasates throughout the body, except for the central nervous system, the brain. At the same time, the brain is uniquely metabolic in that it serves the high demand energy needs of the brain. So for one example here, if you inject FDG glucose analog, you can see it accumulate at very high levels in the brain. And so you already see this kind of paradox, right? You have this barrier property, and yet you can see incredible transport processes. And so then it's no wonder that if you have dysfunction of the blood-brain barrier, you're likely going to have neurological consequences. And so one example here is if you just take a pie graph, right, of folks who are cognitively healthy versus those who have dementia, the takeaway is that more than half of those with dementia have some sort of vascular pathology, either primary vascular or it's mixed with classical Alzheimer's disease pathology like amyloid plaques. And so this is a very big denominator in all dementia cases. So what's going on, right? I think the takeaway for me, at least, is that the blood-brain barrier, contrary again to its namesake, is not just some monolithic structure. It's a very sophisticated community of cell types that dynamically regulates brain health, right? And today, the focus will really be on this first part. Hey, what are the cell types that make up the BBB? What are their organizational principles? How do they talk to each other? And how does it go wrong in disease, right? And so today, it's really about generating this first molecular map of the human BBB. And a question I get a lot is like, okay, yeah, that's fine, but why is this even interesting? And to me, it's because the BBB is like a second half of the brain. You've got at least 6 or 7 cell types that reside here, right? You've got the endothelial cells, pericytes, T cells, perivascular macrophages, astrocytes, fibroblasts. And even historically, people have recognized this concept, right? If you kind of look at how vessels and nerves trace each other in parallel, all the way from Vesalius to today, if you do immunofluorescence, you can see that there is this interesting parallel between, for example, neurons and the vasculature. And so I think it's important to start figuring out, how does this other half of the brain work, and how does that contribute to brain health, right? And so just a quick overview of what we know about the vasculature in the brain. We know it follows the same principles, high level, that the peripheral vasculature follows. In the sense you've got arteries become arterioles and capillaries, postcapillary venules and veins, all these terms, but each of these has a specialized function that they execute. And to mediate that kind of specialized functions, you have different cell types with different cues and molecular bases in each of these segments. And what's unknown up to now is for the blood-brain barrier, okay, how is it different then from the peripheral vasculature? Because we know it's very different. What's the molecular basis of that difference? And then the second part of the question is, okay, we know human brain is quite evolved and different from that of a mouse, right? What's different about human blood-brain barrier? And so the gap in the field has been the following. Since 2015, we've had an explosion of papers doing single cell, single nuclei of the human brain, especially postmortem brains, right? And you can just look at some of the recent high profile papers. And we're now over 200 of these papers a year if you just kind of go on PubMed. But what's interesting is, for reasons that are not really clear, if you were to do the normal prep from human brains, you would basically not capture the cells of the blood-brain barrier, all the cells that I just outlined, right? For example, you can capture a minuscule amount of endothelial cells, 1%, but we know there's a severe depletion, because if you just look at some of these density studies in the brain in situ, you know the vasculature cells should be as represented as all the glia combined, right? So what's going on? It ends up being a somewhat simple issue based on just kind of the physics of how we process brains, right? We take a brain tissue, we put it in a douncer, we mash it up, and then we put it on a gradient, and then we put it on a strainer and add it to our 10x controller or flow sorter, right? But what happens is when you do this downcing, you break up most of the cell types into a single cell solution, but you don't for the vasculature of the BBB, right? So you end up getting these little microvessels, and you can even see the nuclei in them. They kind of look like, bump on a log morphology. But then when you try to clean it up on a strainer, like you have to do to avoid a clog, right? You just kind of lose them like you lose spaghetti on a strainer, right? And so then the idea is, okay, let's just turn the strainer around. And then the second issue is, okay, we have these guys in high purity. How do you get the nuclei out? And so, like I mentioned, I studied engineering, and there's a tool called a pea viner. I ever wondered how you get peas out of a pod? There's this dedicated machine to do that. And we just took inspiration from that, miniaturized that. And so that you can get nuclei out of these vessels without damaging the nuclei themselves, right? And so we call this technique VineSeq. It's an acronym for vessel isolation and nuclei extraction. And when you do this, you get a very different kind of set of cells captured, right? Now you get the cells of the blood-brain barrier. So in green here, for example, the brain endothelial cells, you get the pericytes, smooth muscle cells, the fibroblasts, perivascular and meningeal. And you also get the perivascular macrophages and T cells, right? And so if you just compare that to prior studies, you can capture at least 10,000x more of a given vascular cell type. And some of them were just not captured at all before in the human brain, right? And so even before we get into some of the science, the major takeaway is, hey, if you're doing human brain, single cell, single nuke, maybe you want to consider doing VineSeq if you want to capture this other half of the brain, right? Okay, so just a quick run through of some of the biology. First, what we can do is we can start decoding, what are some of the principles by which the BBB is constructed in health, right? What are the cell identity markers? What's different between a mouse and a human? And we have this term called zonation I told you about, arteries, capillaries, veins. Hey, what's the molecular basis for that? Then you can go into, hey, how does this change in Alzheimer's disease, potentially contribute to what's been known about, blood-brain barrier breakdown or leakiness? Well, that's causation, and that's correlation. And so can you start getting to causation, right? And so can you start asking, okay, which cell types express some of these Alzheimer's disease risk genes, and can you validate that, and what does that mean? Okay, so just very briefly, principles in health, right? Just giving 3 examples, right? So the first is, now we can capture perivascular macrophages. We also capture microglia. It's a lot of interest in, hey, what is the difference between these 2 myeloid compartments, right? And so you can just do this kind of typical DG analysis, and all this data now is on our online browser, and it'll become available, all the raw to process data. But you can recapitulate some known findings, for example, from the Butowski lab, that TGF beta signaling is important for microglia. But there's a lot more that folks in the field can unpack with this resource now. Another example is, okay, what's different between mouse and human, right? So we can, for example, do VineSeq on aged mouse brains, similar with aged human brains. And you can just see in this kind of scatter plot, most genes are kind of similar in their expression on diagonal, but you can see genes that are uniquely expressed by species. And you can do this now for all the different cell types and validate them. But when you do that, what's interesting is you see that brain endothelial cells seem to be the most evolutionary divergent among all the other cell types. So brain endothelial, then microglia, and then pericyte, right? So that's something that we'll circle back to when we talk about Alzheimer's risk genes, what that means. We can talk about zonation, right? If you kind of double click on just the human brain endothelial cells, you can capture the known zones and you can validate that with established markers. But you can then now expand that and say, okay, in an unbiased way across almost 700 genes, what are their patterns, right? So when folks want to validate in tissue, oh, hey, what's a human artery marker? Well, they can just look through now and do that. Or what we're having discussions with right now with Jackson Lab is, hey, let's make a brain endothelial specific Cree driver, right? Now we have a set of markers to do that. And one interesting takeaway is for the kind of zonation. You also see that that's species specific, right? I mentioned to you earlier how brain endothelial cells are very divergent. You can say, hey, within a vein, right? Which genes are specific to a vein for a mouse versus which are those specific to a human in a vein? And you don't see a lot in the diagonal, which is interesting. So that means, for example, I'll just show you one example. VWF, von Willebrand factor, is a very important gene when it comes to thrombosis and the risk for stroke. And it seems to be very specific to just the veins in the mouse brain vasculature. But in humans, it's ubiquitous, it's everywhere, even in the small diameter capillaries. And we know that the greater the expression of von Willebrand factor, the greater the risk for ischemic stroke, right? And so this could be one reason why it's been so tough to translate mouse stroke models. But we're still trying to work out, hey, what does this exactly mean? And there are many other genes like that, where it becomes scrambled in the zonation pattern. Just very briefly on, kind of what goes wrong in our Alzheimer's disease patients, right? We have about 10 controls in Alzheimer's disease, and we can just start asking what's different about the AD patients versus the controls, right? And we know, for example, that, like I showed you earlier, vascular pathology is very common in Alzheimer's disease and neurodegeneration. And one motivation for this is, there's a big area of research right now when we do single cell and looking at what are specialized cell populations of microglia in Alzheimer's disease, right? These disease-associated microglia, or DAM. And it's been extrapolated even beyond AD now to where you say, hey, maybe there's a DAM cell type for all these various diseases, and maybe even development or normal aging. And so then the simple question is, okay, given that transcriptional plasticity that you see in microglia, what do you see in the vasculature in Alzheimer's disease in humans, right? So again, you can kind of just color code our patients by control or Alzheimer's disease. And right away, what's very interesting, at least to us, is that you see not an emergence of a new cell type in Alzheimer's, but you see a loss of these cell types, right? So across the board, you see a loss of the vascular cell types like the endothelial cells or the pericytes. And for the endothelial cells, what's interesting is it's kind of consistent across each vessel segment, right? So, that kind of comports with people's notions that, oh, you get these what they call string vessels where the endothelial cells die out and you're left with just the basement membrane. And so this seems to be ubiquitous across the zones. What's interesting for the pericytes is it seems to be selective. When we had done this kind of single cell, we are able to subcluster the pericytes because we capture so many of them, about 30,000 of them. And what we noticed that there are two cell types of pericytes. One is, mediating transport processes, small molecule solute transport. And the other seems to be responsible for constructing ECM, the basement membrane that kind of creates the structural integrity of the BBB. And in Alzheimer's disease patients, we see a very selective loss of just these matrix making pericytes, right? And so that could be one molecular basis by which this kind of BBB breakdown that we see in Alzheimer's disease patients, by which that works, right? Here's a potential molecular mechanism. So that's something that we're hoping to explore further. And then finally, you can do this kind of DEG analysis, right? And if you do that, you can kind of see, oh, for the pericyte and smooth muscle cells, something that stands out a lot is you see kind of what you'd predict to be kind of constricted or lessened blood flow. So you kind of see increased cell contraction, negative regulation of blood flow, and that also fits in with what David Atwell and others have reported. If you expose these kind of mural cell types, pericytes and SMCs, to amyloid, you get that constriction. And that's what we're seeing in patients' brains. Okay, so that's correlation though, right? So you can't really say, okay, maybe the Alzheimer's pathology came first and then you saw these effects. What about the other way? And this part is still tentative. We're still working it out. But let's start with the motivation. The motivation is that up to 80% of Alzheimer's disease risk is genetic. A lot of them through to non-coding SNPs, right? And the pipeline to kind of interpret these SNPs has been, all right, let's go, fine map them and then let's go assign a cell type to them, and then let's link a gene in that given cell type. And so that way we can start validating this either in animal models or in iPSCs, right? And a big cell type in the Alzheimer's field has been in microglia, and rightly so. I mean, there's been a beautiful set of papers showing that microglia express many of these risk genes. And interestingly, it seems to be different in mouse versus humans, right? So in humans, microglia, they seem to express more of these Alzheimer's risk genes than the mouse microglia counterparts do. And so the simple question that we had is, okay, well, if you do your conventional prep, you capture these kind of 5 typical cell types, right? Microglia, astrocytes, oligos, OPCs, and neurons. And what if we just order now the top 45 Alzheimer's disease risk genes, starting from ApoE, that everyone knows, all the way down, and you just color code it by expression level, right? So if you just saw this half of the brain, yeah, microglia definitely express most of these genes. We should definitely go after them and study the mechanisms. But now if you kind of use VineSeq and expand it, you start seeing, hey, wait a minute, there's actually quite a bit of expression in the vascular compartment as well, right? So you see quite a bit in the endothelium, in the T cells, and even in the perivascular macrophages that are the kind of the myeloid counterpart to microglia, some of this expression exceeding that in microglia. And you can validate this in situ, right? And so, for example, we just show you here 12 examples, but most of these you can validate, including, for example, ApoE, right? We think it's mostly microglia and astrocytes, but in humans, we see it also around smooth muscle cells. You can kind of double click on some of these genes now and ask, like I mentioned to you earlier, like, hey, what's the difference between mouse and humans? And you can see, for example, in endothelial cells, these risk genes are almost always higher in human endothelial cells than in mice. And they map to protein transport or immune functions. Here's just another kind of representation of that. I talked to you earlier about ApoE and smooth muscle cells, and you can see from a mouse data set, it's not expressed at all in mice. And then you can generalize this, right? Beyond just the 40 that I showed you in that heat map, there are something like 600 that people have linked to either Alzheimer's or AD related. And almost all of them are higher expressed in human than in mouse BECs, right? And that's not true though for the whole transcriptome, the 20,000 genes. And so this seems to be a very selective evolutionary shift, where all of a sudden the vasculature, the BBB cells are expressing more of these Alzheimer's risk genes for somewhat unclear reasons right now. You can ask, okay, for these like 680 related risk genes, which cell type expresses the most of each one, right? And you can see, oh, hey, absolutely, the myeloid cells express a lot of them, but so do the endothelial cells, right? And if you do pathway analysis, you see very interestingly, they reside in these kind of ligand-specific receptor-mediated transport processes, right? Clathrin, Golgi receptor complex. And so that fits into this model that we have now, which is, yes, microglia are still the central players in mediating response to Alzheimer's disease pathology. No doubt about that. But maybe, given the dramatic evolutionary expansion of the human brain, metabolism, for example, amyloid beta is a metabolic byproduct, you need more auxiliary support for the microglia. And that support comes from a lot of a transfer of these, Alzheimer's risk genes towards the endothelium, even perivascular macrophages and T cells, right? And so this is an area that we want to study a lot more. We're basically protein communication, immune cell communication across these barriers, how they contribute then to Alzheimer's disease. And just very briefly at the very end, I just want to highlight what we think is going on here when it comes to kind of protein transport across the BBB and linking that to a prior paper we had in Nature in 2020, showing that there is an unexpected degree of BBB transport. And we think this is kind of worth highlighting because, up till now, again, the namesake of barrier has been deduced by injecting one tracer at a time, right? All the way from this classical study with Evans Blue. And if you kind of do that, you're led to believe that there's minimal protein transport because there's no vesicular transport, there's no vesicles through the brain endothelium. And we had instead flip the script and say, hey, people are doing CRISPR screens now to be very unbiased in their discovery. Why not do something similar with chemical biology? So let's not tag one thing, but let's tag 1,000 things at once, right? So in this case, we just took plasma proteome, thousands of proteins, and optimized how to tag them in a non-denaturing way. And when we inject that labeled plasma into a brain, a mouse brain, you see, the vasculature is lit up with these puncta, with these vesicles, signifying transport. And we could indeed validate, for example, that neurons are taking up a lot of this signal, right? So it's starting to converge where you see a lot of these Alzheimer's risk genes that mediate protein transport in the human brain endothelium, but also you can see that, hey, there's a lot of plasma protein transport. And so hopefully, next steps, these two worlds meet, right? Where we want to, for example, be more conclusive in linking these non-coding SNPs to these risk genes, starting to validate that with using CRISPR in a dish, but then also starting to decode that language of communication in mouse models by developing new tools, right? Bioorthogonal chemistries, cell tracing, and so on. And maybe one day these two worlds can meet, right? Where we start realizing, oh, maybe these protein transport genes in brain endothelial cells contribute to Alzheimer's disease risk because they enable a protein to enter that doesn't belong. Or maybe now a protein that should be cleared is not being effectively cleared, right? And so really it's about developing new tools and bringing it back to Alzheimer's disease biology. And so we just started 2 weeks ago. We have a team of 3 RAs and students now, and we're always looking for great postdocs to join us. And with that, acknowledge great group of mentors at Stanford, team of RAs at Stanford, and happy to answer any of your questions. Thank you.
That was terrific. Thank you very much, Andrew.
Sure. Thank you.
Jennie has to leave earlier for an emergency meeting. So, so now we're open to the floor. If you have questions, feel free to unmute and speak up, or you have questions for Rohan, then now it's the time for you to ask. Okay. So before I see any questions, someone's wondering what are the percentage of the AD patients in your samples that have CAA, the cerebral amyloid angiopathy? I thought that the presence of CAA might affect the BBB a lot.
Yeah, in our case, actually, only 2 of ours had notable CAA to a neuropathologist. Most of them, though, did have some type of ferritin accumulation, which is another type of vascular pathology, but not a lot of CAA. The next steps for this, it's going to sound crazy, but this is kind of collaborative work with Tony. We are now processing 150 of these using BindSeq. And in that case, we're going to have a whole mix, right? From controls to pathologically, a little bit of CAA to a lot of CAA to stroke to full AD. And so you'll hopefully be able to start linking, oh, hey, phenotype like CAA to transcriptomic data.
Thank you. And the last part of your talk is something you would take with you to your new lab, right? Yeah, I haven't been reading something about the blood-brain barrier permeability assays. So could you just elaborate a little more about the new techniques we're developing?
Yeah, absolutely. So very simply, what we want to do is we want to create a first catalog of what proteins and cell types can enter the brain. And the way we do that is by developing new tools. So instead of injecting, let's say, Evans blue or dextran, and one by one, let's figure out a way to tag them in a comprehensive way that doesn't change their biology. So we can use, for example, these kind of bioorthogonal chemistries, and then inject them in, for example, to young or age or disease model mice. And then we can do kind of this enrichment affinity mass spec. So basically, it's like a fishing experiment. You tagged your proteins with a special handle, you wait for them to enter the brain, and then you can fish them back out and wash away all the endogenous protein or cell in the brain tissue. And then you can do mass spec to identify what those are, right? And so hopefully by doing that in a comprehensive way, you can start looking and seeing patterns, right? Like, hey, we see this type of protein, or maybe all the proteins that enter the brain have this peptide motif, or it's these cell types and they enter because they have this type of receptor, or they enter through this portal. That's what we want to get towards.
Very cool. And you say you're recruiting technicians, postdocs?
Mostly postdocs, yeah. We already have
okay.
3 RAs and students, and so now it's looking for postdocs, yeah.
Nice. Okay. Good luck.
Thank you.
Okay, any questions from the participants?
Hi, Andrew. Nice talk. I do have a question for you. Have you ever tracked different brain regions for the endothelium cells? Like some brain region maybe have more plaque, or like whether brain region affect the analysis as well? Thanks.
Yeah, absolutely. In our single cell data, we had hippocampus and cortex, and you see very different results. So for example, the hippocampus, even if you're looking at just the control patients, normal age, no amyloid pathology, we see increased inflammation in the brain endothelium, and we see already a loss of pericytes. And so it suggests that the hippocampus is selectively vulnerable, even in normal aging, outside of kind of Alzheimer's disease context.
Thank you. I have another question is that, do you see any ApoE status affect the result as well? And also, are you going to do those experiments by using like ApoE 3, 4, or 2 mice to validate the findings from the human?
Yeah, it's a good question. We see huge effects with ApoE4, and it actually is interesting. It also triggers incredible inflammation in the endothelial cells. Basically, every interferon component gene goes up in ApoE4 versus E3 in both the cortex and the hippocampus. And hopefully that will be helpful for the field to start understanding why ApoE4 patients. I know if you talk to like Betches, Logovich, and others, they think it's a huge contributor to blood-brain barrier impairment. We think now it's due to this tremendous inflammation. In terms of mouse models, absolutely. We're looking at some of these humanized ApoE4/3 knock-ins with Yadong Huang here at the Gladstone, and hopefully we'll see something interesting there.
Thank you.